Welcome to MOOC course on Introduction to Proteogenomics. In the last lecture, Dr. Bing Jiang gave you very lucid elucidation of studying polymorphism. The QTL studies help to understand the effect of genetic variant on another gene located on the same or different chromosomes. GWAS studies help in identifying whether a particular gene variant from the entire DNA set could be correlated to a variation in the phenotype. In this lecture, you are also introduced the single nucleotide polymorphism or SNPs and genome-wide association studies or GWAS studies. Today's lecture from Professor Bing Jiang will be another effort to explain the power of integrating expression quantitative trait loci study or EQTLs with genome-wide association studies or GWAS. So, let us welcome Dr. Bing Jiang for today's lecture.
So, let's look at some examples. The first example is this group. So, basically they know this SNP is associated with hemoglobin concentration in blood cells, and then but they don't know why this happened, and of course we are interested in how this could happen, right? So they did EQTL analysis, and through the cis EQTL analysis they found this SNP, this exact SNP has a good association with the gene expression right next to the SNP. The gene is called the SMIM1. So, and then you can generate a hypothesis that the SNP probably affects the this hemoglobin concentration through this gene expression, right? But that's just a hypothesis. But at that time, there is nothing known about this gene. But what they did was to, through some gene network analysis, I'm going to talk about it later in the next lecture, and then they found this gene is particularly associated with the hemoglobin metabolism genes in the network, indicating it might have the similar function in that process. And then through some functional experiments in human and also in model organisms, they demonstrated indeed, and this gene is involved through deletion experiments. We found a causal relationship between this gene expression and the phenotype you are interested in. So this shows the power of doing the cis EQTL analysis. And also, we can think about the trans EQTL analysis. Let's say this SNP is associated with systemic lupus erythematosus or SLE, this disease. But at that time, nobody knows how this SNP is associated with that disease. And through EQTL mapping, they found that this SNP is associated with multiple genes, and one gene is C1QB, and with decreased expression of C1QB, and also increased the expression of multiple genes involved in the type 1 interferon response pathway. So this helps them to think that maybe this SNP has an effect because it has through the alter the expression of these genes. And the interesting thing is that the decreased C1QB and increased type 1 interferon response has been a hallmark. It's already known the disease has this phenotype. So, but now we know which genes are mediating this impact. And similarly, through the traditional GWAS analysis, there are multiple SNPs that has been associated with type 1 diabetes. But again, we don't know how these SNPs execute their effect. But through EQTL mapping, this group found that although these SNPs are located at different locations of the genome, but they converge, so they alter or they control the expression of the same genes. That means different alterations, genotypes converge to the same gene expression alteration and eventually change the phenotype. So this shows, examples basically show how you can integrate the GWAS analysis through getting the DNA sequence and also the, for example, RNA-seq to get the gene expression and combining them, and of course the phenotype, and then you will be able to not only associate the SNPs with disease phenotype, but you also know what are the gene expressions that are involved in this. You would think, I mean, the protein expression is also important, and we now know that the protein does not necessarily perfectly correlate with gene expression. I mean, if you do this at the protein level, and you probably will also get, I mean, additional new insights, right? But because of the technology is, I mean, less matured or lagged behind the RNA-seq, those technologies, there are relatively fewer pQTL type of analysis. But this audience, I think we should think about this approach in order to integrate the GWAS study, for example, with pQTL studies. And actually, there are some groups started to do this. And in this study, so basically, they look at the 75 lymphoblast cell lines, and then they did the genome-wide genotype study and the RNA-seq ribosome profiling through RiboSeq and the SciLec proteomics experiments. So now you have the genotype, you have the MR expression and the ribosome occupancy and the protein expression. So, and they were able to identify they focused only on the cis eQTL, RQTL, and pQTLs, and they found hundreds of and sometimes thousands of these cis QTLs. And then they asked, I mean, if I found a cis eQTL, what's the likelihood I'm going to find the same like RQTL and pQTL, meaning the SNP controls the gene expression, also the MR expression, also the ribosome occupancy and the protein expression. That means consistent, right? But they found, for example, if you look at the RNA, of course, RNA is one, but only 88% of the RNA, the eQTLs can be replicated in the RiboSeq experiment or the RQTL level, and only 67% can be replicated at the protein level, the pQTLs. So this basically indicates not all the eQTL effects are reproducible at the pQTL level. Also, it's the same, I mean, if you have a protein QTL, only 35% of them can be observed at the eQTL. That is kind of consistent with our observations in the MR and the protein expression are not perfectly coordinated, but this also indicates that, for example, SNP might have effect only at the protein level, but not necessarily at the RNA level. This could happen, for example, if the SNP is in a region that is controlling translation of the protein rather than the transcription of the gene. So, but we can look at these examples. For example, in this case, we can see the effect is consistent at the protein level or the ribosome occupancy level and RNA level, because the three genotypes, the effect is kind of colored by three different colors. We can see the difference can be observed at the RNA level, the ribosome occupancy level, and the protein level. But in this case, we can see the effect is only observable at the protein level, but not at the ribosome and the RNA level. That means this SNP is affecting the translation of this protein or maybe the stability of this protein without affecting its RNA and ribosome translation, ribosome occupancy. So this indicates if we do both eQTL analysis and pQTL analysis, it will give us more information than just doing the eQTL analysis. But for this study particularly, they only look at the cis eQTL, they didn't look at the trans QTLs. Because as I said, when your sample size is small and the trans eQTL is less difficult to observe because the effect is relatively smaller. And finally, I want to show one example. And what we have talked about so far are all about SNPs, which are the germline alterations, right? Because this audience is interested in cancer, so we can borrow the same idea and apply to the cancer studies. And in this case, we didn't look at the SNPs. We look at the somatic copy number alterations, because in cancer there are a lot of chromosome regions that are getting amplified or deleted, right? And then we can consider that as the genotype change, right? And then we want to ask whether the genotype change will affect the gene expression, mRNA expression, and the protein expression of that gene, or maybe it will affect other genes in the genome. So, and for this, we did analysis in 90 colorectal tumors, and then we calculated the copy number alteration for individual genes based on the SNP array data. And then we get the RNA-seq data, so basically we have the mRNA abundance for each gene. And we did the neighbor-free shotgun proteomics in this study and got the protein abundance for the genes. And we only focused on the genes have both mRNA and the protein measurement. This correspond to close to 4,000 genes at that time. But for copy number, we have the data for 23,000 genes. And now for each copy number data and all the mRNAs, we can calculate. But in this case, the genotype is also continuous, right? Because it's a copy number measurement. And then we can calculate a Pearson correlation between the genotype and the mRNA expression. And if there is a significant positive correlation, we put a red dot here. That means the copy number change of this gene will affect the mRNA abundance of that gene. And then if there is a significant negative association, we put a green dot here. And if there is no significant, we leave it blank. And this is a plot we get from this analysis. And the interesting thing is we observed two types of interesting patterns. One is at the diagonal, we see a very strong diagonal pattern. And the other type of pattern is these stripes, the vertical lines. Let's say if we use this gene as an example, this locus as an example. If this copy number amplification will increase the mRNA expression of the gene in that locus. So you are going to have a significant copy number change and the mRNA expression change. And then you are going to get a red dot, because here all the genes are ordered based on the chromosome location, and here it's also based on the same chromosome location. So if it's a cis effect, you are going to see a red dot here, right? But let's say this gene is a transcription factor, and the DNA amplification of the transcription factor not only cause a higher abundance of that gene, but this transcription factor will in turn activate or deactivate a lot of other genes. So then you also see a genome-wide effect of that DNA copy number change. So we call this the cis band or on the diagonal, because it's the copy number change that will alter the mRNA abundance of the same gene. But we also see the genome-wide, these vertical bands, these are the transband, meaning copy number change at this position may affect a lot of other genes in the genome. And then we also look at the protein data. So here is the correlation between copy number and the protein. We can see both the cis and the trans bands getting weaker. We get kind of similar patterns, but it's weaker. This indicates not all the impact at the MR level can be carried over to the protein level. This is called the phenotype dampening, meaning there is a reduce of the effect. For example, if the copy number amplification gives you more mRNAs of a certain gene, but this gene does not give you the growth advantage of a cancer cell, the protein is not needed, then we don't need to make that extra protein. So that means there is additional regulation at that level that will remove those effects that is not necessary. So, and we also sometimes see the copy number protein correlation only observed at the protein level, but not at the corresponding MR level. This indicates that copy number change might only affect the protein, especially for the trans effect. So this is very helpful, because now we can look at this plot and we can see, okay, there are certain chromosome regions that have particularly strong impact at the global level. For example, this 20q region seems to have a big impact genome-wide for mRNA and protein, and there might be some interesting genes in that chromosome. And we also can look at the trans effect or cis effect, and we can see this indicate the large circle indicate all the protein coding genes in this 20q amplification region that we have both mRNA and protein measurements. And this indicates the genes with the good copy number and MR coordination, and this indicates the genes with good copy number and protein coordination. So this helps us to narrow down to the genes that the copy number will not only increase the MR level, but also increase the protein level. And these are the very likely cancer driver genes in the region. And for example, through this approach, we were able to identify SRC, which is a very well established oncogene in colon cancer. And then the TUM34 has also been reported, and we were able to identify a new candidate driver HNF4A, which could be a new discovery to be tested in the future. So just to give a quick summary of the talk. So we talk about the genotype and phenotype, and using association study to understand the relationships or test the relationships, and depending on whether it's a binary trait or quantitative trait, we have to use different types of statistical tests. And if we do this at the genome-wide scale, it's called a GWAS study, and we can use Manhattan plot to visualize the results. And then if we treat the gene expression as a quantitative traits, and then we can do the eQTL, RQTL, pQTL type of analysis, and these QTLs can be divided into the cis eQTL and trans eQTL based on the positional relationship between the SNP and the genes. And we showed a few examples that you can integrate GWAS and eQTL, or eQTL, RQTL, pQTL, or copy number and the MR and the protein expression to understand the relationship between the genotype and gene expression and the phenotype. Maybe we can have one or two questions, or maybe we can discuss during the lunch.
Copy number analysis, right?
Yes.
Can you please open that? In that, the last column, so can it be like the mRNA degradation, it's not getting degraded, and which is one of the reasons most of the proteins are stable. I mean the transcripts are stable.
So I think it could be but if you think about the RNA-seq, right, it's actually measuring the steady state mRNA abundance. So I think the degraded RNA will not be measured at the RNA-seq. I mean, if you consider that measurement is at the steady state, I mean, that has already been taken care of. But of course, I mean, it's kind of dynamic, so it might partially reflect that effect. But I would think that most of the mRNA measurement we have is for steady state, so that it's already incorporated both the RNA generation and the degradation has both been measured in that measurement. But also, I mean, this is not the abundance of the mRNA or protein, right? It's association between the copy number and the mRNA or protein. But I think the discrepancy is more likely to be caused by the altered translation or the protein degradation, the half-life of the proteins. Yeah.
Today's lecture broadly explained the use of various integrative analysis to understand disease pathobiology. Using examples from published literature, cis eQTL and trans eQTL mapping integrated with GWAS study were shown to correlate the gene expression with phenotype. Additionally, the integrative analysis of somatic copy number variations and mRNA abundance were seen to be directly correlated. In the next lecture, you will be introduced to the next generation sequencing technology and its application by an industry application scientist. Thank you.